persons to please introduce Dr. Neeraja.
Morning everybody. Now we have moved on to the very interesting topic for all of us. To deliver a lecture on this topic, Dr. Neeraja, who is a immunologist at Apollo Hospital. She's a life member of Indian Association of Medical Microbiologists, and she, to her credit, she has a gold medal, and she has won many accolades, academic achievements, awards for research papers as well. And she's a member of Indian Women Scientists Association, Indian Society of Histocompatibility and Immunogenetics, and is a member of Apollo Hospital's Transplant Authorization Committee. Over to you, ma'am.
A very good morning, everyone. Thank you, ma'am, for such a nice introduction. First of all, I would like to thank the Department of Nephrology, NIMS, a place where I worked for 20 years. And now I am coming back to give a guest lecture. It's an honor for me. So the topic of my presentation today is HLA typing, will the increasing resolution solve the problems? So HLA was called from 1967 and recognized as a very polymorphic system. So let us start with the current understanding of two unique classes of proteins in our body, the ones that have the highest degree of polymorphism, and these are unique as most of all other proteins are nearly identical between the humans. So HLA typing is a crucial step in renal transplantation, as recognition of the foreign HLA by recipient T lymphocytes would trigger an immune response against the allograft. So I don't want to go deep into this topic as Dr. Sirapani has already discussed. So the first organ transplantation was done in 1959 by Joseph Murray and his colleagues in Boston. They have successfully transplanted a kidney that was donated by the fraternal twins, and it functioned for 20 years without immunosuppressive drugs. And during that era, I think a kidney surviving for 20 years is a big thing, as the technology was not advanced as it is now. So why the organ transplantation is difficult? It is difficult because of the HLA antigens which are located on the cell surface. Now HLA, also referred to as MHC, plays an important role in intercellular recognition and discrimination between self and non-self. So this MHC is basically present in the short arm of chromosome 6, and its essential role lies in control of self-recognition. So the HLA is comprised of three separate clusters of genes: HLA class 1, that is A, B and C, and HLA class 2, that is the DR region, DR, DQ and DP. And these are highly polymorphic and polygenic. So the basic rule of HLA inheritance is that there is a set of 6 HLA groups: A, B, C, DR, DQ and DP, and this set is known as haplotype. So as per the law of genetics, any person, there is a basic rule, like if there are two siblings, there is a chance of 25% full match between the siblings, and there is a chance of 25% no match between them, and 50% chance of sharing one haplotype with your siblings, as shown in this picture. So as I said, they are highly polymorphic. They show the highest diversity in the allelic variants within the human population. So among the class 1, HLA-B is the most polymorphic, and in class 2 it is HLA-DR, which is most polymorphic. Now this polymorphism basically lies because of the variation in the peptide binding cleft. And as you all know that HLA-DR, when we are denoting it, we write it as HLA-DR B1, B3, B4 and B5, and this variation is because of the beta chain of HLA-DR. Now when we write for HLA-DP and DQ, we write it as DP-A1 and DP-B1, DQ-A1 and DQ-B1. So this is because of the variation in the alpha and beta chain. So as you can see in this slide, there are a number of variation in the alleles of HLA antigen. So I will discuss in details about the types of resolutions which will lead in the discovery of these new alleles. And as shown in this slide, the serological HLA specificities, like for example, HLA A24 belongs to serological group 9, and then A29, 30, 31, 32 and 33 belong to serological group 19. So that's how we write a report when we are doing a reporting of HLA typing. So what happens in HLA typing? So in this test, the donor's antigens are expressed on the surface of the leukocytes of the genes. They are matched with that of the recipient. So the closer the match antigens on the transplanted organ, the more likely that the recipient's body will not reject the transplant. So value of HLA matching between donor and recipient varies in different solid organ transplantation. While in kidney transplants, there is a substantial benefit if all the polymorphic HLA alleles are matched. So as per the collaborative transplant study, which antigens are more significant in typing? So HLA-DR and B antigens, as I said, they are highly polymorphic. They offer the most alloimmune burden with less so from HLA-A. That means if there is HLA-A mismatch, it doesn't matter immediately, but if there is HLA-DR and B antigens mismatch, it leads to lot of antibody mediated rejections and all. So influence of HLA-DR mismatching had the most effect during the first 6 months post-transplant, while the maximal effect of HLA-B mismatch occurred 2 years post-transplant. So data from the United Network for Organ Sharing Registry further highlighted the significance of paying attention to having the least number of mismatches for a successful transplant. So as per the UNOS, when the first adult kidney allografts from a deceased donor, if it is having a 6 HLA mismatch, there is a 64% risk of transplant failure. But if there is only one mismatch, the risk falls to 13%. So now we should understand how a HLA nomenclature looks like. Is there any point? So when we are so when we are writing about the HLA, so HLA-AA is the gene and followed by an asterisk, that is the separator. A02 is nothing but the allele group and followed by the HLA protein. Then the third field is used to show a synonymous DNA substitution within the coding region, and the fourth field is used to show difference in a non-coding region. So this is how a HLA typing, when we give the report with next generation sequencing, with the highest resolution, it is in the 4-digit. So HLA typing methods are generally characterized at low, intermediate and high resolution depending on the power to discriminate between the HLA alleles. So if it is a low resolution typing, it is only till the antigenic level and the result composing the first field of HLA nomenclature like A01 or A02. So if the typing is with intermediate resolution, which is done by a DNA based typing result that includes a subset of alleles sharing the digits in the first field of their allele name, and that excludes some alleles sharing this field. If it is done by high resolution, it is defined as a set of alleles that specify and encode the same protein sequence within the antigen recognition site. So this level of resolution resolves all the ambiguities resulting from the polymorphisms located within exon 2 and 3 for class 1 loci and for exon 2 for class 2 loci. So all the HLA typing, they generally cover exon 2 and 3 because that is the region where the polymorphism happens, and in the exon 2 of class 2 loci. So all the kits should target exon 2 and 3 to make the typing more specific. So the HLA typing is basically done by 3 methods. Serology, although it was a gold standard, now it is being superseded by the molecular techniques as they become more robust and time efficient. Now cellular method of HLA typing is rarely used because originally it was used only for class 2 typing, and molecular is fast becoming the method of choice by most of the laboratories. So first I will discuss about the serological method of HLA typing. The basic principle is it is done by using the pattern of lysis of peripheral blood lymphocytes in the presence of complement and typing the sera to type serologically defined HLA antigens. So lymphocytes are HLA typed by cross-matching to panel reactive antibodies using the complement dependent cytotoxicity. Now if there is a, like the cells are viable, that means the cross match is negative. And when we do typing, if the cells die, they take up the vital stains like eosin red or trypan blue, and when they take up the stain, that means the cells are dead and that particular antigen is positive. So the cells are examined under a phase contrast microscope and cells that are pink are positive. So if 60% or more of the cells are stained, they are considered positive for the HLA antigen. But there are a lot of pros and cons of this method. The pros are they are easily performed and does not require expensive equipment. It takes only 3 hours to perform this test, but the resolution is low level. With good anti-sera, you get a reliable results. But the cons of this test is that you require large volumes of blood because you have to extract number of serological antigens from different types of population like Asian, Hispanic, Caucasian or African. Then the another drawback is you require viable lymphocytes. So we cannot do it from a sample which is being transported to our laboratory or to our hospital from remote places. There will be self-death among the lymphocytes, and hence this is a big backdrop. And it is very difficult to find good anti-sera for rarer antigens in different populations. So the second method is serological typing by using bead arrays like on flow cytometry. Here the recipient anti-human antibodies are assessed by cross-matching to known lymphocyte antigens conjugated to microparticles, and the results are assessed by flow cytometry. I don't think like this is not followed by most of the laboratories. So I don't want to go deep into this. So now the third method is molecular typing method. So the three basic methods in which HLA typing is done is sequence specific primers and sequence specific oligonucleotide probes. This is the Luminex method, which is being done by almost all the laboratories in India. And the third one is sequence based typing, or that is called SBT, and this is next generation sequencing. So what happens in the molecular typing methods? There are 3 steps of molecular typing. The first step is to extract the genomic DNA from the whole blood, that is the EDTA blood which is collected from the patient and the donor. Then we have to extract the DNA. This is followed by amplification of the genes of interest and detection of the sequence polymorphism that define the alleles. So these are the 3 basic steps of molecular typing. So based on the classification of resolution, as I discussed earlier, the typing region for class 1 is exon 2 and 3, and for class 2 it is only exon 2. So when they design the primers for typing, they will design from only these 2 regions for class 1 and class 2. So the genomic DNA, when it is extracted from the sample, as you know that DNA is present everywhere. So you have to make sure that the DNA which is extracted is highly pure, and the purity of the DNA is checked by biophotometer, and there is a parameter called A2 absorbance at 260 by 280, and that should be between 1.8 to 2 to ascertain that your DNA is pure and you are safely going for HLA typing. So the second step, as I said, gene amplification by PCR. So the specificity of the amplification can be locus specific. For example, HLA A, B and DRB1, that is group specific. DRB1 O1 or O2 is allele specific, and DRB1 O4 O1, these are all allele specific primers will be designed. So for PCR, the specificity is determined by the sequence of primers and the amplification conditions. So most typing scheme require conditions that avoid the co-amplification of pseudogenes, because in PCR the most important problem is contamination. So there should not be any amplification of the pseudogenes that will interfere in the HLA typing. So amplification of exon 2 for class 2 is sufficient to achieve the highest resolution level, and for typing class 1, both exon 2 and 3 are amplified by a single pair of primers. The first method of HLA typing, as I said, is the sequence specific primers. So what does PCR SSP stand for? It is polymerase chain reaction amplification with sequence specific primers. Now if the target sequence is present, the amplicon will be generated representing a positive reaction. As you can see in this gel picture, there will be control amplification of the genes, and wherever there is a band below that control, that means that particular gene is present. So the amplified products are analyzed by agarose gel electrophoresis, and the pattern of positive and negative reaction is used to establish the HLA alleles. But this SSP typing is highly expensive. It is labor intensive because the more number of genes, the more electrophoresis you have to do, and it is a very tedious procedure, and you require large number of primers to do PCR SSP. So the most interesting part of HLA typing is PCR SSOP. That is the basic principle of Luminex, which is present, I think, in all the laboratories all over India, whoever is doing the transplant workup. So here the basic steps, as I said, is DNA isolation and generic PCR amplification. Now the most important or crucial step here is the denaturation of the bioattenuated product. That means in the first step, when you amplify the gene, the HLA gene, that has to be denatured, and then a bioattenuated product is added. So probe hybridization with group or allele specific probes, binding and identification in the Luminex. So it provides high throughput testing with less sample handling time, and can deliver the results within 4 to 5 hours of typing for HLA class 1 and 2 antigens. So this is how Luminex looks like. So HLA typing using Luminex is a reverse PCR chain reaction sequence specific oligonucleotide system, which involves PCR amplification of target regions within the MHC class 1 or 2 with group specific primers, followed by a process of probing the amplicon with Luminex beads, each coated with sequence specific oligonucleotide probes to identify the presence or absence of specific alleles. So once the beads attach to the Luminex thing, the assignment of the HLA type is based on the reaction pattern observed compared to the patterns associated with the published sequence. That means the Luminex software already has a database of the already published sequences, and when we do typing of patient and donor, it will go and match to the already existing sequence, and then it will give the result as HLA A1, O2 or O1 and so on. So this is how we get the results in Luminex. So the advantages of Luminex for HLA typing is the combination of speed and reproducibility form the main advantages of Luminex as used in HLA typing. The technique is fairly robust. It requires very little DNA. We are only with around 2 to 3 ml of whole blood we can do HLA typing of either patient or donor, and fewer staff is required for this, and reduction or even elimination of the use of agarose gel electrophoresis and its associated use of ethidium bromide, which is considered to be a little bit carcinogenic. But the main disadvantages of Luminex HLA typing is the kits are little bit costlier, and you get the results in low to intermediate resolution. Hence, you require further testing by sequence-based techniques. So, it is in practice that when you are doing a renal transplant, the physicians or the clinicians, they ask for HLA typing of only HLA A, B and DR. Is it right? Yeah, I think most of them, they ask for HLA typing A, B and DR. But in Apollo hospital, we found a case where there was antibody mediated rejection due to DQ antibodies, and this was the first report from our hospital. So this was a 28-year-old male CKD patient who presented to our hospital for cadaveric transplant. The patient has a history of 3 units of blood transfusion, and he came to a hospital on 20th February 22. So when we did a pre-transplant workup for this patient, the CDC cross-match was negative. Luminex DSA for class 1 and 2 was also negative. So we did, it is basically a practice in a hospital to do single antigen bead also for deceased donors, especially if the patient has a history of sensitization. So single antigen bead class 1 was negative with a PRA of 0%, and class 2 was positive with a PRA of 1%. And he showed the presence of DQ A1, O1, O2 and O6, O4 with a very low MFI value of 1836. So we discussed and we said, okay, fine, the MFI is 1836, so it is not so significant. So let us go ahead with the transplant. So the transplant was done on 21st February, and the patient was on regular follow-up as per our protocol, like once a week for 2 months, then once in 15 days and so on. Then after, when he was going on with regular follow-up, the recipient was presented on 12th May with a very low urine output. So there was a suspicion of antibody mediated rejection. So now on 19th May, the single antigen bead class 2 was repeated, and to our surprise, what we found that the PRA which is reported in single antigen bead, it raised from 1% to 16%, and the MFI, the same antibodies were detected even after transplant with DQ A1, O1, O2 and O6, O4, and the MFI had increased from 1800 to 5832. So we thought we will let us do a virtual cross match. So for virtual cross match we did, since we already preserved the DNA of all the cadaveric donors, because it is difficult to get the transplant, the sample in the later stages. So we took out the DNA of the cadaver donor and we did HLA typing, and we found that the virtual cross match was positive. So this slide shows the single antigen bead report of the patient, the one which is marked in the circle, and when we did the donor HLA typing, to our surprise, even he was having HLA DQ A, O1, O2 and O6, O4 antigens. So based on this data, we came into the conclusion that the virtual cross match is positive. So the patient was treated with IVIGs and 2 cycles of plasma exchange was done, and after a few days the patient was discharged in a stable condition. So the take home message from this case is, please include DQ typing also in the pre-transplant workup, because DQ is responsible for antibody mediated rejection in most of the publications recently. So HLA typing by NGS here. So as per the latest release in January 23, it contains 35,820 allelic variants. So with the increase in technology, the number of alleles which are discovered has also been increasing from low, when we are going from low to high resolution typing. So next generation sequencing is a massive parallel sequencing technology that offers high throughput, scalability and speed. The technology is used to determine the order of nucleotides in the entire genome or targeted regions. It offers to detect novel or rare variants with deep sequencing, and 4 fields of resolution is achieved by NGS as it resolves the ambiguity in exons, thereby achieving better tissue compatibility in transplant. So we did a study of around 150 renal transplant patients. We did NGS, and when we, I don't want to go deep into the steps of NGS. The basic steps are, you collect the sample, you extract the DNA, it is subjected to long range PCR, quantification of the amplified product by qubit. This is followed by fragmentation, adapter ligation and library preparation and sequencing, and finally the data analysis. So after the data analysis, we identified many novel and rare variants, and we submitted these rare variants to the GenBank and further to IPD IMGT HLA database. So this is how the results of NGS looks like with a 4-digit resolution. Like you can see in this slide, HLA A, O3, O1, O1, O1 and so on. So here also I would like to discuss just one single case. we identified a novel allele called HLA B44256 in renal transplant patient from West Bengal in eastern India. He was a 48-year-old male CKD patient with a spousal donor. And there was no family history of CKD or hypertension or diabetes in his family. So high resolution HLA typing by NGS was done, and the slide shows the HLA genotype, and we identified this B44256 in this patient. The nucleotide sequence, we suspected that generally the B44 is denoted as B44O20101, but this was B44256, so we submitted to the GenBank, and we found that the DNA sequence of B44256 differs from 44O20101 in exon 3. As I said, exon 3 is the gene of interest in MHC. So there was a variation in exon 3 at multiple nucleotide positions, and this was submitted to GenBank, and this data was published in HLA Immune Response and Genetics. Since this novel allele was identified in this patient, we suspected that this gene would be inherited to his daughter also. So we advised further HLA typing of his daughter, and this was also seen in his daughter. So we identified one more novel allele, HLA C12O2O222 in renal transplant patient, and even this was published in HLA Immune Response and Genetics. So to conclude, I would say that following implementation of the NGS methodology, we have discovered many novel HLA alleles with direct implications for donor selection, as well as many novel alleles with intronic differences. Identification of the novel HLA alleles by high resolution HLA typing lead to less ambiguities and better transplant outcome for patient. Full-length gene sequencing and the ability to curate full sequence data will permit future interrogation of the impact of introns and expanded exons and other gene regulatory sequences on clinical outcomes in transplant. The novel allele which is detected today could be a new one, but in future that could be associated with any of the diseases, because HLA is also associated with many diseases like B27 for ankylosing spondylitis and so on. So the use of high resolution complete HLA genotyping significantly improves the correct identification of donor specific antibodies both prior to and after kidney transplantation. So to conclude, which HLA typing method is best for renal transplant? Currently the standard of care for HLA typing is to select a donor for transplantation to use with a low or intermediate resolution first. And if there is a match, we can type the eligible candidates using high resolution to confirm the results and select the best match donors. Thank you for your patience listening.
Thank you, Dr. Neeraja. An excellent presentation, including the methods that you told and also clinical cases presentation also you made. But for just discussion for the postgraduate, how we are using this technology? If at all a patient with live liver transplant coming to your OP, or a cadaver transplant is coming to you for selecting the donor. Why we are discussing this method is to select the best donor. So are we using for selecting the best donor in these two scenarios, live and cadaver? Live, what we are doing in the OP, we are only checking the blood group. Then we'll also check the donor profile, like age, number of blood transfusion that the recipient received, and the age and diabetes or comorbid condition of the donor. Generally, HLA nowadays are being used to determine the relationship only, to tell you frankly. We are using HLA typing for determining the relationship between the donor and recipient. We are not using this to select the donors, because if at all you have plenty of donors, then you can have this technology to select this donor is better, that donor is better. And apart from that, you also use this HLA typing for determining the immunosuppression. Two things. One is what is available best donor. Second is what immunosuppression you have to use post-transplant. That whether we are using optimal in living donor, no. As of now, as clinicians, we are using HLA typing for only determining the relationship. If it are the haplo match, it's okay, they are relatives. Not haplo match, we are trying to still further thing. That is the thing. In the cadaver also, what we are doing cadaver, we are using index, kidney donor profile index, KDRI. That is a better determinant in accepting the donation from the donor or not. We are not checking the HLA at all. This kidney donor profile index doesn't have HLA in it. It is not factored in when we calculate the KDPI. So definitely availability of the technology is important, but what is the relevant for clinical practice also we should be able to make it clear. So if at all you have multiple donors, then you want to select a best donor among them, definitely this is useful. If at all blood group is matching, everything is matching, like profile of the donor is matching, then you want to use a better donor among them, definitely you can go for HLA typing. That also what madam suggested is high throughput, or like high resolution, what we call. Now HLA A, B, C, D, only we have 2 antigens, but up to 6 levels also, 6 digits also we can go in. Like in NIMS also we have. Now NGS is also there, and the PCR based is also there. But we rarely use as a clinician. We only depend for HLA typing for determining the relation. That is the clinical relevance of this technology which is there. But definitely the technology, we need to accept the progress in the technology and also use for the betterment of the patient outcomes. So whenever you have donors in plenty, in that scenario this is useful. And it also, if at all you want to have a tie-up in let us say in the cadaver donation, you have all the scoring is matched, but based on HLA you want to give a better kidney. That also a lot of studies are there. Actually the KDPI is more important than the other factors. So relevance is still evolving. In future, I think if at all the further, because the number of the immunosuppressive drugs have improved, which improve the kidney survival in a transplant, and also other factors playing a role. So relevance of this need to be discussed in a research setting and also clinical relevance. So Dr. Vijay Kumar.
So that's a very good presentation. So you're talking about NGS. What is the cost of blood testing when you're covering exon 2 and 3, right?
But in NGS it is a whole genome sequencing. For class 1 it will be covering the entire genome, and class 2 it will be covering only exon 2 and 3. So what we do is NGS, as everybody thinks, is not so costly, because we have a chip technology in NGS and a flow cell. So in that flow cell you can do around 96 samples together. So the more the number of samples, the cost will be less. So actually the cost per test is only 2,000 to 3,000 when we are doing for NGS. And in corporates or in governments, the cost per test, you add more value to it. So it generally what we charge is 6,000 to 7,000 to the patient. But the only thing is the turnaround time when you do for NGS is 1 week, because the sequencing itself takes 3 days. It takes around 18 hours when we introduce the amplified product into the Illumina. We have Illumina ISEC 100 in our Apollo. So once you do the sequencing and load the cartridge into the sequencer, it takes 17 hours for the sequencing to happen. So we keep the TAT to 7 days, because you need to pool the number of samples. We cannot do NGS for 1 or 2 patients. Around 20 to 30 is the best option to go ahead with the NGS. And as Dr. Gangadhar has said, HLA typing is okay for donor selection or to establish the relationship. So what we always try to convey to the clinicians is, even if the patient and donor are unrelated, we can do the HLA typing of patient and donor and keep the record with us. And when you are suspecting antibody mediated rejection, we can do single antigen beat for the patient, and we will be having the donor typing result with us. So at that point you can do virtual cross match and find out which are the antigens which are against the donor in the patient, and based on the MFI you can tailor your treatment. Like if the MFI is around 10,000 or 20,000 like that, you can plan for the plasma exchange, and the dose of immunosuppressive drugs is fine, but in the later stages it can be oncogenic also. The higher the dose, the patients go into, they are suspected to have lot of other infections like CMV or EBV and all. So in order to prevent these post-transplant infections, if we do HLA typing of both patient and donor and keep a record with us, it will be very useful for post-transplant monitoring. So that is what I wanted to convey as immunologist.
Thank you, ma'am. Lucid presentation. Thank you very much. Any questions from the floor?
we are entering to the virtual cross-match era. Even it is true for disease donor transplant. What Gangadhar told is a practical thing that we are not using beyond the sort of, proving the relationship. But that's the older concept. The newer concept of when you are trying to do virtual cross-match, doing the HLA, even in disease donor, do the HLA details analysis before and for the recipient, and then quickly you can do the donors before transplant, or even if you do not have the time to do it, or sort of facilities and a lot of logistics, you can do it, keep the blood, do it later on, and then draw a conclusion, which she was mentioning. So there is the immense role of HLA, which is coming back in a different way.
I request all the chairpersons and speakers to be brief in their question answer because we are running short of time. So,
Yeah, I agree with Dr. Their role is definitely there, but in what context we are using, that's the important thing actually. So, and only for the benefit of PGs I told, but if at all we have resources, definitely I like to sequence all the people in both the donors and recipients and have a bank. Based on that, yeah, that also will be done, but only the cost constraint and all those things to be worked out. So where we can have a bank based on this, that also can come. But practically, what is happening, I just told. So any more comments from the floor?
so actually in disease donor also we are not breaking this HLA for this matching or the allocation, but we are trying to preserve this donor sample and get this HLA done subsequently whenever we want to go for whether we have this the rejection and we have to look for the DSAs. We are preserving the donor sample. In fact, we have started doing the HLA typing also for the donor recently.
Thank you, ma'am. Thank you for that informative talk.